So the next topic is cell fate. So earlier I have already discussed what is cell potency. So cell fate is nothing but the different cell types that the cell can become, or the different descendants that it will produce during a normal development of the cells. So you can understand that cell potency is the ability, as I have already discussed, the ability to give rise to different cell types of a particular cell. So the cell fate is nothing but the end product of this ability. So what will happen is that cell fate will be equal to cell potency, but in presence of certain environmental factors. There will be always some external factors which will prevent the cell from becoming all the possible outcomes it can become. So the equation is given like this, that cell fate is equal to cell potency plus environment. Some factors will enhance the potency and therefore give rise to many fates, and some factors will hinder the potency of the cell and will give rise to lesser number of cell fates. So cell potency is therefore greater than equal to cell fate. It will always be either equal to, like whatever is the potency of the cell, exactly that will be the fate, or it will be greater. The potency will be greater, and due to some external factors or environmental factors that has decreased, and therefore the ultimate cell fate is much lower than the potency of the cell or the actual ability of the cell. So you can understand that as a cell progresses from a totipotent cell to an unipotent cell, the cell fate and potency, they both are decreasing. So its fate is actually getting restricted. So because the number of cells it can get differentiated into are becoming fewer and fewer, and therefore its fate and potency are getting restricted or decreased, as you can say. So the fate of a cell can be described as the future identity of the cell, or whatever is the identity of its daughter cells, before it is actually detectable through differentiation or division, because you are assuming what it can become. So before actually seeing what it is becoming, you are assuming that it will become this type of cells. And cell fate can be determined by three factors, mainly the endogenous developmental factors, like whatever is there inside the cell will determine to some extent what it will become. Interaction with adjacent cells, the environment comes into play, and the adjacent cells might influence that particular cell to become something else or the same type. And external long distance signals such as morphogens or hormones. You know that signaling molecules like morphogens or hormones are very important role players in these types of events. So if some signal comes which favors the cell to develop in some other way, then the fate can change. And in general, if you see a stem cell, it can go through multiple pathways. Like first, it can go through a cycle. Like in case of symmetrical cycle, it can form two stem cells of the same type, which is known as self-renewal. Or it can form two cells of different type, which is known as differentiation. Like I told you, the production of a specialized cell type is known as differentiation. Or it can go to an asymmetric cycle and give rise to one stem cell which is of the same type as its own, and one differentiated cell. So one of its own kind and one a new kind. Or it can move into a quiescent stage. Quiescent is basically an inactive stage. It does not multiply, or it does not show any activity, but still it is alive and it is surviving. Or it might go into apoptosis, which you know is a programmed cell death. It might move into the death phase. So these are the possible cycles it can take, and when it takes the self-renewal cycle or the differentiation cycle, then you get to see different kinds of cells it can produce, and thereby the fate or the cell fate comes into action. So next, when I move on to is commitment. So this is a very important term in Dev Bio. So commitment is basically the cell, when the cell is it has a particular specific fate, and it will differentiate into that particular type only. Then you can call that the cell is committed to that fate. There are some other terms in commitment, like the naive cell is when the cell is not committed, the uncommitted cell, if you can say. It has no definite fixed pathway which it will follow. So we can call this a naive cell. And from here, when certain cytoplasmic determinants or inductive signals play their role, you can see that the naive cell becomes a specified. What is specification? When a cell is committed to a given fate, but this commitment can still be reversed, like from the specified cell line, like from when the specification has already occurred, it can be reversed back to the naive stage. So you can understand that this specification is not permanent, but it will differentiate along unless signals or its environment changes. So if there is another signal, then it might reverse back to the naive stage, or if there isn't any such signal, then it will move on to being determined. What is determined? Like, determination is the commitment to a given fate which is irreversible. So here it cannot reverse back to its naive stage. When it's determined, that means it will definitely move into that pathway and form that particular cell type, or whatever is its fate, it will fulfill that. So the cell will differentiate along a given pathway regardless of its location. So you can see how a specified cell can move into a determined cell by loss of competence for alternative fates. So what does that mean? It does not have the competence to go into some other fate. That is why we are calling it determined, which means it is determined to one particular kind of fate. And from here, once it is determined, it will move on to differentiation. What is differentiation? No, it's like a formation of a specialized cell type. So at this stage, the cell is overtly of a certain type at a biochemical and functional level. So like I already told you, a specialized cell type is nothing but when it is showing a completely different function, as well as a biochemical reactions are all different compared to their parents, like the parent cells from which it is produced, then we call it a differentiated cell. So you can see that as a cell becomes committed and progresses through specification, determination, differentiation, its fate progressively decreases or gets restricted more and more. So it cannot form some other cell types. So the important thing here is that for commitment, there is mainly two factors. One is the cytoplasmic determinants, which is an intrinsic factor of the cell itself, and the other is the external signals, like the inductive signals which it will get, so that the cell, these actually help the cell to decide which pathway it will follow and exactly which fate it will choose. So the next is cytoplasmic determinants. Like I told you, they are special signal molecules, nothing but present in the cytoplasm. That is why they are called cytoplasmic determinants in the female gametes, which are responsible for the regulation of gene expression in the early development of the embryo by differential specialization of blastomeres during cleavage. So what happens is that there are certain factors or molecules present inside the female gamete and in the cytoplasm of the female gamete, and their unequal distribution results in some cells getting the majority of these determinants and some cells getting very less amount or not at all. So what will happen is that when these cells which have received the cytoplasmic determinants, they will move on to form something else, and these cells which are devoid of any cytoplasmic determinants, they will move on to a different fate. So their fate which is determined will be different, and in this case their determination will be something else. So this is how the cells are committed to a single fate by unequal distribution of the cytoplasmic determinants. So at first, August Weismann gave a germplasm theory, which means that there are nuclear determinants in action, and the theory, he hypothesized that during first cleavage these nuclear determinants get unequally distributed, and in second cleavage they are getting, you can see that each cell has a different type of nuclear determinant. But later on this theory was abolished because you already know that the genomic content of each cell of a particular organism will always be same. So these nuclear determinant theory is not valid. So later on it was then proved that cytoplasmic determinants are playing a major role in this part. So what are the types of specification? Autonomous, conditional and syncytial. These are the three major types of specification. The first two are more important. Like autonomous specification is mostly the characteristic of invertebrates, and conditional specification is the characteristic of all vertebrates and a few invertebrates. So how does this happen? So autonomous specification is nothing but the specification by differential acquisition of certain cytoplasmic molecules present in the egg. So basically whatever I explained just now by cytoplasmic determinants, that is the specification, autonomous specification. So it produces a mosaic or determinative development cells cannot change fate if a blastomere is lost. So this means that their specification is so determined that it produces a mosaic. This kind of development is called a mosaic development. Why? Because if you remove these cells, then these cells containing no cytoplasmic determinant can never give rise to the fate or the cell types that were supposed to be produced by these cells, the bottom cells. So if you if you detach these cells from the main embryo body, then you will find that the embryo does not have those cell types which these cells were supposed to produce. So this kind of development is called a mosaic development. And in case of conditional specification, it is specification by interactions between the cells. Like I told you, adjacent cells play a very important role in case of induction, and therefore this kind of specification is mainly governed by inductive signals. So I will go into inductive signals in the next slide. So what is this kind of development called? This is called regulative development. Why is that? Because it allows the cells to acquire different functions. So even if in case of inductive signals, even if you remove certain cells which those cells were supposed to produce, you will see that the other cells have acquired the ability to produce all the possible cell types even when they are missing, and then give rise to an entire complete organism. So this kind of development is known as regulative development, and it is a conditional specification, like it is conditioned like that. But in case of autonomous specification, it is already fixed. The determination is already done such that it cannot be changed, it cannot be altered, it cannot be influenced by something else, but it is fixed. So this is the basic difference. And syncytial specification is nothing but when it is a characteristic of most insect classes, and this specification is mainly done by a multinucleated cytoplasm. So syncytium means a multinucleated cell or something like that. So from here you can understand the specification of body regions by interactions between cytoplasmic regions prior to cellularization of the blastoderm. So before cellular division or cytoplasmic division occurs, the nucleus gets divided, and that stage is called a syncytium because it has a multinucleated stage. So in that stage there will be interactions between different cytoplasmic. So in the entire cytoplasm only there are different interactions going on, and based on that the development will progress. So I will just go into this slide so that you understand better. So in syncytial specification you can see that this is nothing but a Drosophila egg that I am showing. So there are two factors which are present. One is bicoid and one is nano. So you can see that these are present in two different poles of the egg. So what happens is that when there are nuclear divisions, so you can see there are number of nuclears present here, and according to that the bicoid and the nano starts progressing in its opposite pole, towards its opposite pole. So what happens is that there is a gradient, like here the concentration of bicoid is higher and compared to here it is lower. Also in the same way, just the reverse is happening. The nanos, the concentration is higher, and here this way it's becoming lower. So after cellularization, what will happen is that the entire cell cytoplasm gets divided into segments according to the ratio of the bicoid and the nanos protein, and then it gets it is decided like what it will form. This portion will form the head, then this is the thorax portion, this is the tail portion. So this is decided based on the comparison or the relative ratio of the bicoid and the nanos proteins. So this is known as a syncytial specification. So this is how the cell fate is specified in this case, because of the interactions of different cytoplasmic portions of the same cell. Now, in case of autonomous specification, like I told you, like these cells, these are supposed to form a trochoblast. Now, these trochoblast cells, when they are isolated, they are forming the same cells which they were supposed to form when they were inside the embryo. So they are forming the same cells. But if you remove these cells and isolate it, then the embryo will not have these cell types, and other cells will also not produce these cell types. So this is known as a mosaic development. And in case of this conditional specification, you can see that this specification is how, because you are in the blastula, if you remove certain cells like transplant, if you so if you remove the cells from the back tissue, then no transplantation or normal development. So you can see that normally the back has developed. And if you transfer these back cells to the belly region, like cells of the belly region, then the cells will form the belly tissue, and the back will also be formed normally. So when these cells are totally removed, then also you can see that a normal organism is developed. So what does that mean? That the adjacent cells have played the role here. The adjacent cells make them or give them the ability to become whatever cell type it is missing, or whatever cell region it is being transferred to, and therefore the entire organism comes into action. So this is known as a regulative development. These cells regulate themselves and try to fulfill all the cell types that the organism requires. Next is induction. So what is induction? Basically, it's a process where one group of cells and influence the developmental fate of another. So like I told you, like inductive signals are a very important role player in case of commitment or specification. So these are the two ways. Basically, this is an extrinsic way, which is induction. The cells, they signal the other cells and become something else. Or in case of cytoplasmic determinants, like I already told you, there is an asymmetric division because some cytoplasmic determinants move into some daughter cells and some daughter cells do not have any cytoplasmic determinant. Therefore, the ones with the determinants become something else than the ones without. And here also you will see the same thing, that when there are two kinds of cytoplasmic determinants, then also the same thing happens. One daughter cell gets one type of determinants and the other daughter cell gets the other type of determinants. Therefore, obviously this cell will form something else and this cell will form something else. And in case of induction, you can see there are two components, like one is the inducer, the tissue that will produce the signal, and the responder, that the cell which will receive the signal and act accordingly. So in case of induction, this is you can see that the bottom cells are the inducer and the top cells are the responder. So they are passing on the signal from the lower part of the embryo to the upper part, and thereby the cells are getting differentiated accordingly. So how is this signal basically transferred? So obviously, for the signal to be received, you need a receptor. And here also you can see that this cell is basically an inducer. So these are inducer molecules. Okay, so inductive signals as we call it, and they are diffusing, and here you can see that the concentration is higher, here the concentration is lower. So these cells, they will receive lower signals than these cells, which will receive a higher number of inducer molecules. So these are the receptors that are already present in the on the surface of the cells, and you can see that being having the same number of receptor also does not mean that it will receive the same number of inducer molecules because of the concentration gradient. And then when it reaches when the signal reaches the nucleus, and it results in transcription factor activation too, so that those genes which are the target genes, they get activated, and it is getting activated. Here you can see that the protein is encoded and with this protein is responsible for cell division, differentiation mainly. So the cells will be determined to a particular fate, and it will move on to differentiation. But in case of this cell, the number of inducer molecules is less, therefore the signal will not move on by activating the transcription factor. There is no transcription, so you can see that the protein is not produced and the cell does not differentiate.